first of all, thank you for inviting me here. It's a great pleasure, and just having sat through the last 3 talks, I feel deeply, deeply inspired. There are so many important things that we need to do in the world, and I'm really, really honored to be here. So let's talk about Iowa. So I grew up in China. I was born in a city named Shijiazhuang. It's about an hour south of Beijing by train. And I moved to the U.S. When I was 11 with my parents. One of the reasons that my parents decided to stay in the U.S. is because they thought the education system was very different from the one in China. In China, it's all about rote memorization, whereas in the U.S., kids get to explore their own curiosities and follow their passion, and they thought I would benefit from that. In fact, I feel very lucky because had I stayed in China, I'm not a very good person at memorizing things. I probably wouldn't even get into college. So I feel good that I was able to study here. Iowa was a tremendously welcoming community. So as an immigrant, I never felt that I wasn't included in the community. And the school had a lot of great activities for helping immigrants learn about American culture and also to adapt to a new environment. Beginning in high school, there was a program called a corporate academic program. And they were pairing students up with local businesses so that students can learn some vocational skills. And they had a partnership with one of the local hospitals. It was called the Methodist Medical Center. And many students signed up to work in the surgical suite or working accounts receivable to learn different skills that would be useful once they graduate from high school. And it turned out that the hospital also had a gene therapy lab. And my enterprising science teacher said, why don't you try to see if you can get into the gene therapy lab? You might have to wash dishes, but maybe they'll let you do something more. And so I got very lucky. They accepted me and paired me up with a tremendous, phenomenal mentor who really enjoyed teaching and really sort of impressing on me the excitement and also the ability that science has to make the world a better place. And working with him for 2 years, I would go there after school around 2 p.m. every day and stay until 7:30, and just learning from the people there who were working on science and side by side. That was how I got started. And without those mentors, I think I probably wouldn't have sort of such a strong interest for science.
Feng, you talk about the mentors, and that's the what, and you talk about how you got lucky. There's also the why. Going to volunteer for 5 hours after school every weekday is a lot. And some of us in the room already have kids. I'm lucky enough to have a 1.5-year-old. And those of us who don't probably think about maybe having kids one day. And we're all probably thinking, like, how do we get our kids interested in science, technology, engineering, math? What was some of the spark that drove you to dedicate that much of your time?
I think a very large part of it was the mentorship that I got. We all know in school some teachers are better at getting us excited about a topic and other teachers are not so excited. And then there's also probably the upbringing and the things that kids are naturally interested in. I think I probably wouldn't spend 5 hours on playing basketball. I'm terrible at it, and also probably because you're terrible at it, you wouldn't find it so enjoyable. But science is something that I've always found interesting, and having a mentor there who kind of recognizes my interest and then tries to figure out a way to help me develop it, that made a huge difference. So I think mentorship is very important.
So we need to find inspiring mentors for our kids.
Yeah, absolutely.
I want to sort of almost reintroduce you and give you guys a flavor of some of the breakthroughs that Feng has been responsible for. So you started off with undergraduate in chemistry and physics, but then transitioned to the brain, which is really neurobiology. And I know that when you were applying to labs at Stanford where you did your PhD, the traditional neuroscience labs didn't want you because you didn't have a neuroscience background. Mm-hmm. You ended up going to the lab of Carl Dieseroth, who was a bit more of a renegade neuroscience researcher. that ended up being really important in one of the breakthroughs that came out of that lab, which is optogenetics, which is probably one of the biggest breakthroughs we have of studying how the brain works. It's literally being able to shove fiber optic cables into a brain, obviously animal brains, not human brains, and using light to turn neurons on and off. How do you think, having studied different fields like chemistry and physics, helped you to functionalize that invention in the field of biology?
So I guess, so first of all, want to sort of explain a little bit about my interest in the brain. So the brain, intellectually speaking, is very interesting because it's what makes us human, and it's probably the most complicated piece of organ in our whole body. But another experience in college really got me thinking deeply about the brain, and that's because one of my really close friends in college experienced an episode of major depression. Before seeing how depression can really affect someone, I probably thought, like most people, that neuropsychiatric diseases, depression, schizophrenia, bipolar disorders, these are not really real diseases. Everybody can have a bad day, and getting a couple of nights of good sleep, your mood will lift, and then things can get better. But it's really seeing my friends struggle that I began to realize that psychiatric diseases of the mind, are just as real as diabetes or heart disease or hypertension. And the only difference is that we really don't have any way of treating these diseases. Any treatment that we have so far or found by serendipity and not really based on any deep understanding of the disease mechanism. And so that kind of drove me to want to study the brain.
So really tools that could actually go down at a functional level.
Exactly. And because the brain is a pretty big, dense, mushy piece of tissue, we really don't have a way to understand how the brain is made up. It has many, many different cells, and they're all connected to form circuitry that can compute information, allow us to remember things, or to feel things, or to be able to think deductively in a logical way. We didn't have tools to do that. And so thinking about how can you begin to take the brain apart, piece by piece, and figure out what's responsible for, say, how my arm is able to move, or what's responsible for the emotion of happiness or sadness, then we'll be able to start to be able to treat brain disorders.
Yeah, that's amazing. That tool is one of the biggest breakthroughs we've had for being able to actually study the brain and look at these brain disorders, and is now being used by over 1,000 universities and research institutes around the world, just to give some perspective of how a scientific breakthrough can have reach, and by researchers looking at everything from Alzheimer's to depression. And a Nobel Prize will probably get awarded for optogenetics, but that wasn't enough for you, was it? So you got this appointment, you got a postdoc at Harvard, and then got your own lab at the Broad Institute. You were at the time the youngest researcher to have their own lab at the Broad Institute, and actually are still the youngest. Feng is NEXUS eligible. He's 37 years old, and a change maker. You were in just a meeting of visiting researchers, a lot of your colleagues from the Broad Institute around, and you heard this visiting researcher mention this tool that was being used being used within bacterial DNA. What pricked up your ears to make you think that this might be something that was powerful for editing genes in our bodies?
Yeah, so gene editing is about being able to make changes to the DNA in our cells. So we all have a genome. Our genomes are subtly different. Between any two individuals, there are probably about 1 billion different letter differences in the genome. And the genome is 3 billion letters long. And so over evolution or sometimes spontaneous mutations can happen that certain differences in the genome can cause disease, cystic fibrosis, sickle cell disease, you name it. There are about 6,000 genetically defined diseases because we know what differences lead to them. And so if we know what mutations are harmful, one thought is, why don't we go into the cells and then fix them? Replace the mutation with the sequence that everybody has so that we can restore health for that individual. That's what gene editing is. So gene editing, the way it works, is kind of like how you would edit a document. You find the mutation and you put it, or the typo, and you place the cursor there so that you can backspace or delete it and then type in a sequence. So what is the cursor, or the equivalent of a cursor, for the chemical DNA that's in our cells? It turned out that this cursor is a break in the DNA. So DNA is a long string. And if you make a cut in the string, wherever you make the cut, that's where the cell can go and try to repair it. You can give the cell, you can fool the cell into repairing it with a new sequence by giving the cell a piece of artificial DNA. So if you want to fix a letter from an A to a C, you just give the cell a piece of artificial DNA that has a C in it, and the cell can incorporate it into the genome. The way to make a cut is through the use of a protein called nuclease. So nucleases are basically scissors that can go into the DNA and break it. The challenge is, how do you get a scissors to go to exactly where you want it to be? And CRISPR is this system that bacteria has invented to be able to fight off viruses, but we can repurpose it so that we can put it into a cell, give it an instruction to hone in to the typo that we're trying to fix, and then make a cut to stimulate the repair process. And CRISPR, when I heard about it, it was a nuclease system. And because I was thinking about how do you make better nucleases, when I heard that word, that just captured my attention. So I think having that problem in mind, and then comparing the new information that you're hearing with that problem to see whether or not it's applicable, then that's the way that you can find novel solutions to solving a problem.
So I want to get into that tweak that you made to CRISPR. I also want to talk about the third huge breakthrough invention that you made that's going to change the way we live, and maybe briefly touch on the ethics of CRISPR. The clock said 15 when we walked on stage. Are we able to get the 20 minutes, or do we have to finish in 1 minute? Okay. Okay, thank you for letting us have our full 20 minutes. It's such a fascinating conversation. So the CRISPR story is one of incremental advances, and Jennifer Doudna and Emmanuel Charpentier were important people in that story. The breakthrough you made in your lab at MIT and Harvard was to adapt it for mammalian cells. And can you talk to us a little bit about, maybe in layman's language, what was it about your background in physics and chemistry that allowed you to see a way to make that CRISPR tool work in cells that could be mammalian cells, which is all of us and animals?
I think it probably started when I was a little kid. I always like to take things apart, and sometimes I don't put them together, and that makes my parents pretty upset. But I think the interest in taking things apart to see how it works is that engineering mindset that's very useful for tweaking biology. When I first learned about biology, I actually really hated it, because biology taught in middle school is about memorizing things. You get a bunch of different leaves and you figure out what type of trees they're from. Or you're given a smelly frog and you have to take it apart, figure out what a heart is and where the intestines are. And that's all really boring. But when I first really learned about modern advances in biology, I began to realize that biology are built of parts. Our cells have DNA, have RNA, and then they encode genes. And you can tweak these genes to change the way that cells behave. And then if you fix the genes, then you can create therapies, like gene therapy, to be able to treat disease. And so it's that mindset then applied to biology that makes it possible to start to figure out, how does the CRISPR system work? What are the different pieces? How do you tweak each part so that they can work better together and then work in a different environment? It's naturally from bacterial cells, but how do you put it into a human cell and still get it to all work together? Is that kind of taking...
It's like a building box, yeah.
That you can begin to develop technology.
So it's you destroying your parents' electronics when you were a kid, and then the chemistry and physics that enabled you to tackle biology more like a toolbox rather than smushy stuff in a test tube.
Right.
That's so cool. So Feng unfortunately has to leave after this plenary and won't be around for the rest of the conference. If any of you want to be in touch, I can get you in touch with him, or his Twitter is there, and it's a great way to follow what's going on at the Zhang lab at MIT. I want to touch briefly on ethics of CRISPR and then wrap up with the third big breakthrough that's going to change the way we live. So the ethics there, you all heard about the CRISPR baby in China. Who heard about the CRISPR baby in China? Okay, everyone heard about the CRISPR baby. So scientists who invented CRISPR and who still work on CRISPR today all get asked all the time, like, what should we be doing about this? I think it's not really for the scientists in the US to do something about it. It's for us to put pressure on our lawmakers to create an international consensus around it. It was the government of China that probably funded that researcher, and that's not really something that US scientists can stop. But one thing US scientists can do, and I want to ask you, Feng, is for everyone in the audience, what should they walk away with out of this room thinking about the implications? If they're going to speak to their lawmakers and put pressure on our government, what should we be thinking about implications?
So in November, a scientist in China reported that he had edited human embryos and then implanted those embryos back into the mother, and then 2 baby girls who had their genomes edited were born. What he had done was there was no medical justification, the consent process was not appropriate, and then it was done in utter lack of transparency. So the whole process was fraught with ethical problems. And so we call for a moratorium for the use of gene editing to create genetically modified human babies. That's for several reasons. One, the technology is not quite ready yet. And second, the science is not there. We don't even know how to fix many diseases, and therefore we don't know how to do it to really be able to guarantee success in the patient outcome.
And to be clear, this moratorium you're talking about is about germline editing, which means that those changes, those edits will get passed on forever.
Yep.
To the children, the grandchildren, the great-grandchildren forever.
Right. And so there are really 2 ways to do gene editing. One is called somatic gene editing, which is changing cells in the body of the adult. And those cells will not become sperms or eggs, so they can't get passed on to the next generation. The second form is the germline editing. That's the issue that we're talking about here, where you're changing cells either in the embryo so that when the embryo develops into a full person, that person will pass on whatever changes they got to their offsprings, or by changing germ cells. So either sperm cells or egg cells, and those will get passed on as well. So germline editing is an important issue that I think as a society we need to talk about, because the decision to allow gene editing will change the way that humanity works. I'll just give you one example. So there are really 2 types of germline editing approaches. So one is called gene restoration or genetic restoration. This is a little less ethically challenging because we're trying to fix mutations that lead to disease. So the ultimate goal is to be able to relieve suffering and be able to improve human health. The second form of germline editing is about genetic enhancement. So being able to change physical traits, increase IQ points. And these are the things that in the long run, if we do as a society decide to move forward, it's going to cause tremendous changes in the underlying fabrics of society. Some of us are parents here, and we just think about how much effort and mental energy we spend into getting our kids to get into the right school or to learn something. If parents are given the ability to edit their children, to get them to have, say, 20-point higher IQ, then it's going to create such a competitive situation that other parents will be pressured into doing it, because if they don't, their kids will not be competitive in society. Now, not all parents will have equal access to resources, and so some people will have access to resources and other people won't be able to access it. And that's going to exacerbate the inequality that we'll have in society. And so these are just a glimpse of the tip of the iceberg of a problem that gene editing in the germline will create for humanity. And
that's a message to take to our elected officials, that we don't want this accelerating runaway.
Well, so fortunately in the US, law forbids germline editing. But that's not true around the world. There are countries where laws for germline editing is not so clear. And so medical tourism is already a problem. And you can imagine that if there isn't some kind of standard around the world, this is going to become a big problem.
Amazing. It's the whole of our society is predicated on this idea that there's some level of equality and ability to pull yourself up by bootstrings. And if wealthy people can just imbue their kids with more IQ points. That's not going to work anymore. The third huge advance we don't really have time to cover, but I will say that there are a number of people who've been involved in the CRISPR breakthrough. None of them have gone on to do anything revolutionary since then. And Feng, you really impressed me because you then went on and took that CRISPR technology and applied it in a whole different field, not for therapeutic, but for diagnostic. And actually, that's the technology has a cool name to it. It's called Sherlock, easy to remember. You can find out more about it, I'm sure, on the lab's website. But why it's interesting and relevant to all of you in the room is a lot of us care about equal access to medical care in emerging countries, developing countries around the world. And this Sherlock is a tool that instead of editing DNA will let you find any DNA sequence very, very cheaply. So you could use it to find any bacterial sequence or any viral sequence. Think about the implications of that for Ebola or any other mass pandemic outbreak. Is there anything that you thought about for the implications of the Sherlock technology?
I think we need cheap, rapid, and also sensitive diagnostic tests that patients can really do this at point of care or even at their homes or in the field. And right now, the diagnostic technology is not quite there yet. And what we're trying to do with Sherlock is to bring us to that level. If someone has the flu, rather than taking the public transit all the way all along the way spreading the virus before you can get the diagnosis, if there's a kit that existed at home that you can very quickly get a diagnosis, then that's going to be really important for public health. But since I'm on the stage and I have your attention, I just want to make one plug for the Society for Science and the Public. Going back to the very first question, which is how I got interest in science, but it's not just about how you got interest in science, but also about how you sustain that interest. So as a high school student, I was involved in a number of science programs for high school students. Many of them were sponsored by Society for Science and Public. Unfortunately, nowadays, these programs, which bring kids who have similar interests, who may not have as many peers in their local school, but if you just bring different ones together from around the world, they can really start to build a community and also to support each other's interest in science. These programs are now not getting as much support as there should be from different sources, corporations, and also society as a whole. And I think that's another message that I really hope you will take home, which is look at these other programs that are building the next generation of scientists and researchers and engineers, because they are going to be creating things that make our lives even better and are going to solve a lot of these important problems that we face in human health, climate, energy, and many other sources.
Support it and allow your kids to dismantle all the electronics in your house. Thank you for sharing with us today.